Hello, everyone. My name is Bill Curtis. There is a link to my story below. Check it out if you're looking for hope with Parkinson's. The series you are watching is mostly about science. I plan to make several videos that explain how mitochondrial quality control is key to understanding and treating PD. I hope you will get useful information from watching these videos. In Chapter 1, I explained what superoxide and sentinel meant. I talked about steps in mitochondrial quality control. I explained a little about the proposed mechanism that uses superoxide as the sentinel to determine which mitochondria should undergo mitophagy. In Chapter 2, I explained how I got the idea and introduced several people who moved the idea from an off-the-cuff speculation to a well-vetted hypothesis. If you missed any of this, you can find the playlist of all the chapters in the comments below. I promise to talk about how antioxidants work. I want to start with a story. Patrick and I were working on the review comments of a paper. Dr. Veach had invited us to contribute to an article he wanted to publish. I won't say which article because I am going to tell what happened during a review. So, we got back an anonymous comment from one of the reviewers that stated we were wrong to call oxygen a free radical, and we were wrong about NADH and NADPH having different redox potentials. Anyone can look them up in a table. They have the same values. My draft of a comment reply to the reviewer read, "You are wrong about oxygen and NADPH. Once you understand why you are wrong, you will be happy that I told you that you are wrong because these two facts explain so much." Patrick recognized the potential to upset the reviewer and suggested we delete the references to oxygen being a radical and the differences of redox potentials of NADH and NADPH. What did I go on to explain in my response to the reviewer's comment? Well, first let's look at oxygen. Historically, oxygen was the first molecule that had behavior that could not be explained by Lewis dot structures. Oxygen is paramagnetic. What does it mean to be paramagnetic? Michael Faraday showed materials that are not ferromagnetic fall into two categories: those that are weakly attracted to a magnetic field and those that are weakly repelled by a magnetic field. He called the materials that are attracted to a magnetic field paramagnetic, para being a Greek preposition meaning alongside. We now know that paramagnetism is an indication that a material has unpaired electrons. I will put a link to a demonstration that oxygen is paramagnetic and nitrogen is not paramagnetic in the description below. So, oxygen has unpaired electrons. Why was that a problem for Lewis dot structures? Using the rules about valence electrons and placing pairs of electrons into either lone pairs or bonding pairs, there is no hint that oxygen should contain an unpaired electron. This led Leonard Jones to propose that one could make a mathematical linear combination of two atomic orbitals, a LCOA, to get a molecular orbital. Here is a molecular orbital model of oxygen. Let's look at it. To the left is a scale without numbers or divisions that just says energy. So, we can say that the electrons in higher orbitals have more energy than the ones below them. The other major parts of the molecular orbital diagram are the two atomic orbital diagrams and the molecular orbitals in the center. It is missing the 1s atomic orbitals and the sigma 1s and sigma star 1s antibonding molecular orbitals. If you use the rules to fill the molecular orbitals with electrons, you come up with these two unpaired electrons with the same angular momentum vector or spin. Eureka! The LCOA of two oxygen orbitals explains why oxygen has unpaired electrons that can orient in a magnetic field and is attracted to a magnetic field. To be fair, when we look at an organic molecule, we usually use an abbreviated version of Lewis diagrams. This molecule of DHA or docosahexaenoic acid shows how a skeletal diagram is useful for displaying the bonds in a way that we can understand. A molecular orbital chart of DHA would be impossible to comprehend. With a skeletal model, all the vertices are carbons. You need to imagine there is a C at every vertex. You also have to fill in the undrawn hydrogens. Once one understands how the skeletal model relates to the Lewis dot structures, you can see the double bonds starting on the third carbon from the omega end, making this an omega-3 fatty acid. The multiple double bonds make it a polyunsaturated fatty acid. The carbons attached to double bonds are all in the native cis conformation. We just have to know how to view this molecule from the perspective of Lewis dot structures. We see from this it is reasonable for someone in biological science to not have a reason to look at molecular orbital theory's approach to quantum mechanics to explain molecules. Now for the reward. What does the dual free radical nature of oxygen explain? The dual free radical explains what oxygen won't react with. Suppose you wanted to endow oxygen with magical powers to avoid reacting with any potential fuel molecules directly, such as proteins, carbohydrates, lipids, and nucleic acids. You would make it a dual free radical. Yes, that is why we don't just burn up when exposed to ground-state triplet oxygen, because it is a dual free radical. Both of its free electrons have the same spin direction. One of these free electrons must flip to make it react with any of the standard biomolecules. That flip of the electron spin makes it thermodynamically unlikely to happen. If we do add energy to oxygen, we can flip one of the electrons. This takes away the conservation of angular momentum constraint. This is how one ignites a fire, by flipping the electrons in triplet ground-state oxygen to make super-reactive singlet oxygen. The dual free radical explains what oxygen will react with. What will the diradical oxygen react with? Other free radicals. What are the free radicals it reacts with? The iron in hemoglobin is a free radical. Oxygen can bind to hemoglobin because of the iron. Oxygen reacts with the copper in complex 4 of the electron transport chain. Here, oxygen accepts all 4 electrons that have been pulled out of food to become water. Here, 99% of the oxygen is consumed to make ATP without making any reactive oxygen species. Isn't this amazing? How then does the diradical oxygen ever get involved in making reactive oxygen species? To really grasp this, it is important to understand the 3 phases of a free radical reaction sequence. These steps include initiation, propagation, and termination. Once we understand this process, we will also see how antioxidants work. So, let's dig into these 3 steps. Step 1. Initiation. Initiation to an organic chemist is the initial formation of a free radical. This happens when heat or UV light energy is absorbed by an electron. The electron jumps to a higher energy level that is an antibond orbital. This breaks the bond so that you have homolytic cleavage, leaving behind a free radical. The biochemists typically start with a propagation and call it initiation. The propagation they use is the propagation from hydroxyl radical to an R group. The R group donates an electron to the hydroxyl radical, and the R group becomes a free radical. We can take the DHA we just looked at. DHA is a molecule that is susceptible to becoming a free radical. DHA has 5 places in the chain of carbons where bisallylic hydrogens are attached. Here I show just the section of DHA that makes one bisallylic hydrogen. It is an R group, and when hydroxyl radical reacts with the bisallylic hydrogen, hydroxyl takes an electron. What was hydroxyl radical is no longer a free radical. The susceptible region of the PUFA is now a free radical. This is how initiation occurs in lipids. How does one explain propagation and termination? Let's continue using DHA, but only look at this 7-carbon piece that has the bisallylic hydrogens. The reaction with hydroxyl radical made it a free radical. Now we know that oxygen can react with free radicals. When oxygen reacts with the DHA free radical, it forms a peroxyl radical. This is because one of the free radicals on oxygen terminates the free radical attached to DHA directly by pairing up the 2 unpaired electrons. One electron comes from DHA and one electron from oxygen. However, there is a second free radical on the oxygen, and that leaves behind a peroxyl free radical. This peroxyl free radical may pick up an electron from another bis allylic hydrogen. Now we have two funky molecules in the collection of lipids. These could be in a mitochondrial membrane, or in a cell membrane, or in a high-density lipoprotein. What do you think is likely to happen to our new DHA free radical? The DHA free radical can react with another oxygen. The resulting peroxyl radical can make another DHA free radical. This is known as a peroxyl or peroxide chain reaction. Once again, this can only be understood if oxygen is a dual free radical. What is the termination step? Eventually, there are more DHA free radicals, and two of the free radicals collide. They now form a molecular bond that has two electrons. The two former free radicals can no longer propagate. They have undergone a terminating reaction. That does not mean that all the products in the chain reaction are terminated. Just the two that reacted together are terminated. How does an antioxidant stop the chain reaction? Let's look at alpha-tocopherol or vitamin E, which follows the same pattern of free radical propagation. It donates an electron to quench the free radical, such as a peroxyl radical. In doing so, the antioxidant, vitamin E, becomes a free radical itself. The free radical form of vitamin E is relatively stable for a free radical. Vitamin E free radical can get this electron restored from vitamin C. Vitamin C can get its electrons restored by glutathione. Glutathione can get its electrons restored by NADPH. Now we are ready to see why NADPH has a special redox potential in cells. But I am going to save that for the next video. We have come to the end of our video. Stay tuned for the next video where I will tell you all the things you will be able to explain once you know that NADH and NADPH have different redox potentials. Keep your omega-3 supplements in a cool, dark place like a cupboard or a refrigerator, and I will see you there.